All right, hello, welcome back, everybody. Today we need to talk about bacterial genetics and something called operons, which is what bacteria use in order to um, regulate the expression of their genes. Okay, last time we talked about how eukaryotic cells like ours regulate gene expression. So some things are a little similar, but some things are quite different. So let's talk first just about sort of basic bacterial genetics. Okay, bacteria have a rapid rate of reproduction. Okay, and they also have a relatively high mutation rate compared to us. And they also have some ways to do genetic recombination. All of those lead to highly diverse genomes within populations of bacteria. And scientists have used that fact to study lots of basic sort of cellular functions by looking at bacteria, right? A lot of what we know about how our own bodies work comes from the study of E. coli, which is a model organism in labs and has been for a very long time, right? Bacteria make good model organisms for all the same reasons that flies and mice do. You can have a lot of them in a really small space. They're not very picky in terms of growing a lot of them. Um, and because we share a common ancestor with bacteria, right? Remember Luca, the last universal common ancestor. A lot of the ways in which your cell works are exactly the same as what's going on inside of a bacterial cell. All right, just a reminder, bacteria do not have a nucleus because they are prokaryotic organisms. So instead, their DNA is floating in their cytoplasm. The area of their cytoplasm where you'll find the DNA is called the nucleoid region. Okay, the their single circular chromosome will be there with associated proteins. Okay, also because they are prokaryotes, they do not do mitosis or meiosis. Okay, they undergo binary fission. The figure on the right there is showing DNA replication, which again has to happen. Okay, but they have only a single circular chromosome, so they have only one origin of replication and only one replication bubble. And the two forks move away from each other around the circle until eventually you have two complete circular chromosomes that separate from each other. Okay, so most of the um, variation that you find within a bacterial population is going to be due to mutation. Okay, but they are capable of doing genetic recombination as well, right? In humans, eukaryotes like us, um, we talked about genetic recombination happening because of meiosis and random fertilization. Okay, but prokaryotic cells don't do meiosis, so there's no crossing over between chromosomes because they only have one chromosome, right? And they don't do fertilization because that's not how they reproduce. Okay, but there are a couple ways for them to get new combinations of DNA. And a lot of that has to do with what are called plasmids. Okay, and plasmids are separate pieces of DNA that carry information, but they are not part of the chromosome. Okay, so the bacterial chromosome is large and circular, but plasmids are smaller and still circular. Okay, in general, the plasmids cannot function on their own. Okay, so it's not possible to express genes that are part of a plasmid, but plasmids can become part of the chromosome. Okay, so plasmids are kind of a way to sort of store extra DNA that you might not need to use all the time. Plasmids can be made from the chromosome, so part of the chromosome can break off and turn into a plasmid, right? And vice versa. the plasmid can become part of the chromosome, and then it can be used as part of the genome of the bacterium. And the really interesting part about plasmids is that they can be released into the environment and picked up by another bacterium. They can also be transferred from one bacterium to another through viruses, or bacteria can transfer them directly from one living bacterium to another. Okay, so how the plasmid or extra chromosomal DNA gets to the new bacterium determines what it's called. Okay, so transformation, a bacterium is transformed when it takes up new DNA from its environment and incorporates it into its genome. Transduction is when a virus called a bacteriophage or phage transfers DNA from one bacterium to another. And conjugation is when a living bacterium directly transfers DNA to another living bacterium. Okay, so all of these are ways for bacteria to get new genetic information, and when you get new information, you can make new proteins, which allows you to do new functions or build new structures that you couldn't before. Okay, it's actually kind of like if you've ever seen The Matrix, right? In The Matrix, Neo can sort of download new skills and programs into himself, right? In the scene where he goes, oh, I know Kung Fu, right? So bacteria can kind of do that. They can learn Kung Fu during their lifetime just by getting this new stretch of DNA, either from the environment, through a virus, or directly from another living bacterium. Okay, so no matter where the DNA came from, once it becomes part of the bacteria's chromosome, its expression needs to be controlled. Okay, just like us, bacteria need to conserve their resources. Okay, and they need to be able to change how they function based on the environment that they're experiencing. Okay, so they need to be able to turn their genes on and off just like we do. Okay. So last week we talked about how we use activators and repressors that attach to enhancers and silencers, and how we have generalized transcription factors and mediators and those kinds of things that all have to be present in order to turn one of your genes on. Okay, and by that I mean in order for transcription to take place. Okay, in eukaryotes, DNA sections like enhancers can be very far away from the actual coding sequence, right? The part between the promoter and the terminator. Also, multiple genes can respond to the same activator and repressor proteins. Okay, in bacteria, they've sort of condensed that all down into something called an operon. Okay, and an operon can actually group multiple coding genes together under the control of what is similar to a single promoter, okay, or a single enhancer and silencer. All of these parts are brought together into the same stretch of the chromosome in order for regulation. Okay, so a lot of these have similar functions to what we've talked about before, but they might have slightly different names. Okay, so one of the parts that's really important here is a protein called a repressor. And the repressor protein often binds to some other kind of molecule that can be called a co-repressor or sometimes an inducer, and we'll talk about the difference. All right, so every operon has or includes a regulatory gene, and this is the gene that actually codes for the repressor protein. Okay, so this one might not actually be close to the other parts on the chromosome, but it always has to exist somewhere because that repressor protein has to come from somewhere. Just like in eukaryotic cells, every operon has a promoter. Okay, it's a stretch of DNA to which RNA polymerase can attach. Okay, but operons also have what's called an operator. This is a site on the DNA where an active repressor protein can attach. Okay, so an operator site is kind of like a silencer, right? It's a stretch of DNA that something can attach to reduce transcription. Okay, inside the operon there are structural genes, and these are the actual genes that can code for proteins that will change something about how the bacterium looks or functions. Okay, the big difference between prokaryotic and eukaryotic cells here is that in a eukaryotic cell like yours, every coding gene has its own promoter, its own enhancers and silencers. Okay, but here several of them are grouped together because they have to work together to achieve the same goal. All right, so those first four are all segments of DNA. Okay. Next comes the repressor protein. Okay, and this is the protein that can attach to the operator if the repressor protein has its correct active shape. And whether or not it has its correct active shape that allows it to bind to the operator is determined by the presence of a regulatory metabolite. Okay, so that word metabolite just means that it is a molecule produced by the metabolism or needed by the metabolism of a bacterium. So it could either be the starting point or the ending point of some biochemical pathway in the metabolism of that bacterium. Okay, and its presence helps regulate the function of these genes by turning the repressor protein on or off. And it can only bind to the operator when it's on. All right, so let's look at two different examples. There are some operons where you need something from the outside to turn the gene on, and there are some where you need something from the outside to turn the gene off. Okay, and the first example we're going to look at is the trip operon, which is a repressible operon. Okay, one of the things I mentioned in our eukaryotic video was constitutive genes that are basically turned on all the time, right? The cells in your body always need some copies of that protein around, so they're always doing transcription in order to express those genes. Okay, the trip operon is kind of like that. Okay, within the trip operon, the structural genes code for proteins that work together to make tryptophan. Okay, this is an amino acid, which means it makes up part of the structure of polypeptides. You might have heard of tryptophan before. It's the one that is supposed to make you sleepy when you eat turkey. Okay, so because it's a component of polypeptides, right, bacteria always need this amino acid around. Okay, so in general, if nothing else happens, the structural genes in this operon are going to be transcribed. Okay, so these are kind of like constitutive genes. And the operon has to respond in order to turn them off. Okay, so like every operon, it has a promoter and an operator within the DNA, and there is a repressor protein that can bind to that operator. Okay, So most of the time, the bacterium wants this operon turned on so it can make the proteins that help it make tryptophan, which it needs in order to build polypeptides. Okay, but if there is enough tryptophan already present, okay, either for whatever reason it hasn't been using a whole lot of it, or maybe the bacterium found a food source that provided a lot of tryptophan. Okay, so if there is a lot of tryptophan available, then the bacterium doesn't need to make any more. Okay, and under those circumstances, it's better for the bacterium to turn this pathway off and use up the tryptophan that is available before it uses its resources to make more. Okay, so this is what's called a repressible operon. Okay, and what that means is that something has to happen to activate the repressor to turn the operon off. Okay, so let me show you what happens normally. Okay, so normally the bacterium is making tryptophan and using it up just as quickly as it makes it. Okay, so there isn't a lot of extra tryptophan floating about in the cytoplasm of the cell. So without a lot of tryptophan floating around, the repressor protein has an inactive shape. Okay, so the regulatory gene for this operon creates a repressor protein that has an inactive shape. And what that means is it does not have the right shape to bind to the operator. So the repressor protein exists, but by itself, it can't do anything. Okay, so because it can't do anything, RNA polymerase can attach to the promoter, and it can get past the operator and transcribe the structural genes that code for the proteins that work together to make tryptophan. Okay. If the levels of tryptophan get high enough, though, that there's extra tryptophan floating around in the cytoplasm, some of that tryptophan can act as a regulatory metabolite and bind to the repressor protein. Okay, when it binds to the repressor protein, induced fit causes a change in shape, and you now have what's called an active repressor. Okay, when the repressor is active, it can bind to the operator site. Even though RNA polymerase can still bind to the promoter, it cannot get past the operator where the active repressor is bound. And because of that, you won't get any transcription. Okay? So this active repressor has turned the structural genes off, right? So this operon is turned off in the presence of tryptophan. But that's okay because the operon makes tryptophan. So it only gets turned off when there's a lot of product available. Okay? So if you have a lot of tryptophan, you don't need to make any more right now. So you can use tryptophan to activate the repressor and turn the operon off. Okay? So again, if tryptophan is not present, then the repressor has its inactive form and it cannot bind to the operator, which means that tryptophan will be made. Okay? So this is a repressible operon because the presence of the regulatory metabolite turns the operon off. Our other example is what's called an inducible operon. So this is one where the presence of the regulatory metabolite turns the operon on. Okay? So our example here is the lac operon, and it is involved in the metabolism of lactose. Okay, so lactose is the sugar in milk. It is an energy source. Bacteria break it down in order to make ATP, right? Just like you do with energy sources in your body. Okay, so if no lactose is present, you don't need the enzymes to break it down. But if lactose is present, then you do need enzymes to break it down in order to get the energy. Okay, and it has the same name here as it did in eukaryotic cells, right? Inducible genes that only have to be turned on in certain circumstances. Okay, so in this case, without the presence of the regulatory metabolite, this operon, right, the structural genes in this operon are not going to be transcribed. Okay, so we have to turn this operon on because by itself, it's turned off and no transcription is happening. All right, like I mentioned, lactose is a sugar, okay, and it's going to be used for the metabolism, right, to make ATP for the bacterium. Okay, some people are lactose intolerant, right? You can see here lactose is a disaccharide. People who are lactose intolerant, after they grow out of being babies, they stop making the enzyme that breaks the bond between the glucose and the galactose monosaccharides in lactose, right? We've talked a little bit about that. Okay. But bacteria are very happy with any of these food sources. Okay. But they only need to break down lactose if lactose is available. All right, so let's look at this operon. Okay, in the absence of a regulatory metabolite, okay, the regulatory gene for this operon codes for an active repressor. So by itself, the repressor protein can bind to the operator, block RNA polymerase, and keep transcription of the structural genes from happening. Okay, by itself. In the trip operon, the regulatory gene coded for an inactive version of the repressor. Okay, and it only got the active shape when it bound to the regulatory metabolite. But here, all by itself, the repressor protein has the active shape. Okay? If lactose is present, though, a version of lactose acts as the regulatory metabolite and binds to the repressor protein, and because of induced fit, it turns into the inactive version of the repressor. Okay, so when the repressor protein is bound to lactose, it cannot bind to the operator. That means RNA polymerase can get past the operator. It can transcribe the structural genes within the operon. Those genes code for a series of enzymes that break down lactose. Okay, so you only need to break lactose down if lactose is present. And if lactose is present, it deactivates the repressor protein so that you can make the enzymes to break it down. Okay, so the presence of lactose, which acts as a regulatory metabolite, induces this operon or turns it on. Okay, so both of these are types of negative gene regulation. Okay, and they're both considered negative regulation because the active repressor protein keeps transcription from happening. Okay, so in both cases, when there is a repressor protein bound to the operator, which is part of the DNA, transcription is blocked. Okay, the difference between them is that the repressible operon, the TRP one, okay, only has its active shape in the presence of tryptophan, the regulatory metabolite. Okay, so the operon itself gets turned off by the presence of tryptophan. The lac operon, though, it has an active shape all by itself, the repressor protein. Okay, and the repressor protein is deactivated by the presence of lactose. Okay, so the regulatory metabolite here is what we call an inducer. Okay, because it helps induce or turn on this operon. Tryptophan is a co-repressor. Lactose is an inducer. In general, inducible operons are part of catabolic pathways. Right? For example, lactose, catabolic pathways are about breaking things down. So the lac operon creates enzymes that break down lactose. Okay, and you only need those enzymes if you have lactose available to be broken down. Okay, and repressible enzymes often function in anabolic pathways. Anabolic means to build something up. Okay, and you need to build something until you have enough of it. And once you have enough of it, then you can turn that operon off because you don't need to build anymore. But again, both are types of negative regulation. Okay, because when the active repressor is bound to the DNA, right, the operator site on the DNA, no transcription is happening. Okay? All right. Thank you very much.